Good morning, everyone, and welcome to the third session of our spectral.blue Autumn 2024 webinar series. As you might have noticed, your cameras are closed and your microphones are muted to save bandwidth. But please feel free to use the chat for asking questions. We will have a Q&A session in the end of the webinar. The presentations will be recorded and made available for later viewing. So here is the agenda of today. So first we will have some welcome words from our CEO, Dr. Michelle Schmidt. After that, Camilla Höglund, our lead scientist, will tell us how we can replace UV with blue light. After that, I will talk about some customer references that we have. And after that, we will have the Q&A session and discussions. So with that, please, Michelle, the stage is yours.
Thank you, Karin. And a big welcome from my side also. Hey, today is actually my favorite session because 1 hour from now, you will all have a choice. When you go back to the lab and look at your UV and calculate the hours it has been used and reflect about what effect it might have, or you go and test the effect of it, you will have a choice. You can buy a new UV, which is still carcinogenic. It still has mercury in it, so it's not good for the environment. And most probably, it's made in Russia. Or you could make a different choice. You could buy our device. This is totally safe for you. It has absolutely no mercury, so it's environmental friendly. And yes, it's made in Finland. And with those words, I'd like to hand over to our lead scientist, Camilla Höglund. Please, the stage is yours.
Thank you, Michelle, and to all of you, welcome here to our webinar today. I can get the next slide. All right, thank you. So, as Michelle already mentioned, I will talk you through a little bit the differences between UV light and blue light, and a lot of the reasons why blue light is a better option, and in which situations blue light can offer you a lot of advantages that UV light cannot. You already saw the device, one of the devices that we have that is a good option to use in your biosafety cabinets. But we also have customers, for example, in the image to the right, that are using blue light in the whole laboratory. So we have a lot of different options also for installing it in the ceiling and making sure that the whole room will be disinfected properly. First, I will tell you a little bit more about both the advantages with blue light, but also how it works and why it works and why it is a really good disinfection method. I will also, because I think many of you are familiar with using UV light, I will talk you through a little bit about the differences between UV light and blue light and how they can be replaced. And also because we get a lot of questions about the safety aspects of blue light, or is it really safe? Can we be sure that it's safe? I will also touch upon this topic a lot and tell you why we can say that yes, blue light is safe. to this day, we have a lot of customers that are using blue light, as I said, either to disinfect the whole laboratory or disinfecting the biosafety cabinets, or both. One of the main reasons why they would choose blue light is, I think, one of the obvious reasons, that it's safe for people and materials. And I think especially when you're working, you have biosafety cabinets in your laboratory, and you might be working on the bench next to the biosafety cabinet. Would you be using UV light in your biosafety cabinets? You don't want to work next to it while the light is on. But if you're using blue light to disinfect your biosafety cabinets, you can perfectly safe work close to it without having to be worried about any safety issues. The blue light we are using doesn't contain any UV light whatsoever, and I will talk a little bit more about this soon. One of the big reasons for customers to choose this also is the sustainability aspect. It doesn't generate any ozone, and it's mercury free. And I will come back to this topic about mercury a bit later in my presentation. It's also chemical free, and it works really well on biofilm. And this is something I think many of our customers are paying more and more attention to, that there are many situations and ways the laboratories can battle problems with biofilm, and our technology works really well on biofilm. It's maintenance free. If you use our technology for roughly 12 hours a day, we guarantee it to be maintenance free for up to 10 years, as the lead components that we're using have an expected lifetime of roughly 50,000 hours. And this is a lot different to many of the UVC applications, where the UVC tubes would have to be changed every 1 or 2 years. And for laboratories, also the fact that this technology offers a way to do whole room disinfection, continuous disinfection, and it's possible to use without being concerned of the safety issues. You can have it on for longer periods of time. All this will help you prevent cross-contamination and false positives, because the more you can use it and take care of the whole premises and all the surfaces, the lesser the risk of cross-contamination. And also we have customers that have been using the technology in the lab and realize that they can use less chemicals, and they can reduce both the costs of using chemicals and the associated labor of using chemical disinfection. So I will just go through a little bit the spectrum of light and what blue light is and where UV light lies. So here you have the image in which you can see the different wavelengths. And visible light is categorized as 400 to 700 nanometers, and anything below it or above it is not visible to the human eye. So UV light, and especially UVC light that you might be used to using in your laboratories, has a wavelength of 100 to 280 nanometers, and the most commonly used UVC tubes, they have an emission maximum of 254 nanometers. All this is not visible to the human eye. But for you who have used the UVC tubes in your labs, you would know that they look blue to the human eye. And that's correct, because these tubes will emit a very broad spectrum light. And although the emission maximum is at 254 nanometers, they also contain a lot of other wavelengths, and they contain some blue wavelengths as well. And that's why the UVC tubes will emit small amounts of blue light and look blue to you. But if you are using UVC LEDs, they emit only a narrow spectrum of UV light, and they are not visible to the human eye. The blue light technology that we are using doesn't contain any UV light. It contains only blue light, and visible blue light between 400 and 500 nanometers. So how does the blue light work, and how does it kill microbes? Those of you that know UV light knows that it will break down DNA and RNA strands. This blue light technology inactivates microbes in a very different way. So the round circle here that you can see on your screen represents any microbial cell. So it can be a bacteria, a mold, or a yeast. And what we know is that all these microbes contain blue light absorbing compounds inside their cell as a part of their natural metabolism. And these blue light absorbing compounds are present in all microbes, all bacteria, mold, and yeast. What we also know is that when they are exposed to blue light of the right wavelengths and high enough intensity, these are the two key factors. You need to use the right wavelengths and high enough intensity for this technology to work. But when it's working, it's creating reactive oxygen species inside the microbial cell. You can see them here with an orange as ROS. So different types of reactive oxygen species are being formed inside a microbial cell. And as the name suggests, they are very reactive. So they have the ability to break down the cell membrane, DNA, RNA strands, protein structures, enzymes, basically anything that is nearby in the cell. And when this happens, the microbial cell cannot survive. Our technology and our patented invention relies on the fact that we know for sure that there are more than one blue wavelength that works in this manner. And what we discovered also is when you use two blue wavelengths simultaneously, it's much more efficient than using just one. And it's not an additive effect, so it's not like using this and this and you get a summarized or additive effect. You will get the multiplied effect and a many times more efficient disinfection system. Next topic that I want to talk to you about and hopefully get you to understand a little bit better the differences between UV light and blue light. But first of all, one of the factors that I mentioned previously, and one of the reasons why customers are approaching us a lot from laboratories, and especially if you're used to using UV light in your laboratories. It's good to be aware of the fact that all mercury-containing lamps have been banned in EU, and all the white light sources that the traditional gas discharge lamps that we have been used to using indoors, they are already banned. So you're not allowed to produce them or sell them in EU because they contain mercury. But at the same time, the most commonly used UV light source, also gas discharge lamps that needs to be exchanged every 1 or 2 years, they also contain mercury. And they have not now gotten an exemption period that lasts until 2027 because these UV lights are very commonly used in a lot of different disinfection applications, water purification systems. They have now been given an exemption period to find an alternative to the mercury-containing UV light sources. And this is one of the key reasons why many of our customers are now approaching us, because they want to find something else to use. We have, as you saw in the beginning, our CEO, Michelle Schmidt, showed you, we have one device that is very easy to replace and use, replace UV light with and use in your biosafety cabinets. And on our YouTube channel, we have a video also demonstrating how easy it is to start using in your biosafety cabinets. So you just open the sliding window and remove the old UVC device. Our device can be used if you have a magnetic back wall. You can attach it to the back wall with magnets that we provide. But if you don't have a magnetic back wall, we have a solution for that also. So don't worry, you can use it anyhow. And then you just plug in the cord and start using the device. And as I said in the beginning, the key benefits here is that it's safe to use. It's safe to use for both for the staff working in the lab next to the biosafety cabinets, but also safe for all the materials that you might have inside the biosafety cabinet or your laboratory equipment. It works through biofilm and works through most transparent materials. And this can sometimes also be a benefit, that you know that it penetrates most transparent materials, as opposite to UV light, which doesn't. And again, I cannot highlight this enough. It runs for 50,000 hours without maintenance. So it's really just install and forget in that sense. Kind of as a summary a little bit, the differences between UV and blue light. As I said, UV light is not visible to the human eye, but blue light is. Blue light is safe for people and materials, and it works really well on biofilm and can penetrate water and most transparent materials, whereas UV light doesn't penetrate water at all, really. It just reaches the surface. It doesn't penetrate, basically it only penetrates quartz glass, but no other transparent materials. And it's not very, it can have a damaging effect on a lot of materials. Also, this is just for your information. There has, in the recent years, coming out a few studies also raising worries about When you are using UV light in your premises and the UV light's ability to get a lot of air pollutants, different gases and particles to being released from the materials. And actually, this is something many UV light providers might not have paid a lot of attention to, that it can actually, after using UV light, you should evacuate the premises and keep it sealed for some time afterwards, because you will have a lot of unwanted and potentially hazardous particles and compounds in the air a while after using it. This is not something they will warn you about, but there is some studies on this showing that in this particular study, they show that it took 30 to 40 minutes for particle concentration to reach the levels that they were before using the UV light. So just also something to be aware of. Just a few things on the blue light and safety, because we get this question a lot, and there is some concerns, at least here in Finland, there has been concerns raised in the media about blue light hazards and the blue light's effect on eye, and especially when using smartphones and a lot of computers and staring at screens for many hours a day, and this could maybe have a effect on your eyes. All our devices are being tested according to this so-called blue light hazard standard. It's a photobiological safety standard that all light sources need to be tested against to determine the risk group. And our blue light devices range from risk group RG0 to RG2. So there are 4 groups: RG0, 1, 2, and 3. And most white lights would fall into RG0 or 1 category. And they don't require any kind of safety measures. So you're allowed to work under it or close to it for 8 hours a day without any risks to your eye or skin. RG2 devices, we have at least one that has fallen into this category, and that means in our case that it will tell you that if you are closer, for example, closer than 30 centimeters to the device, there's a limit to how long you can stare at it before it's going to cause a risk to your eyes. But if you're further away than 30 centimeters, it's RG1 and no risk. And this is just to be on extra sure. It's good to just pay attention to this also when installing and using the device, to make sure that you don't install it so that somebody can stare at it for a real long period of time. But this is with any high intensity light source, even white light, the same kind of risk to your eyes. You should not stare at anything really close for a long period of time. And I will finish up with just as a comparison so that you kind of realize what kind of intensities we're talking about. There is a publication showing that when you're walking outside on a sunny day, you are exposed to blue light at intensities of between 13 and 50 milliwatts per square centimeters. In addition, you will be exposed to a lot of UV radiation, but now we're just talking about the blue light intensities. And the blue light emitted by our device is typically, when you install it in the ceiling, it's typically 0.7 to 1 milliwatts per square centimeters at 1.5 meters distance. So it's a lot lower. And the table here, you can see if you move really close to this device, of course, the intensities will be higher. But even when you're 30 centimeters from a 100 watts device, you're in the range of blue. It's actually less blue light than if you're walking outside on a sunny day. So having said that, I will hand over to our global sales director, Karin, who will tell you a little bit more about some of our customer references and why they have chosen to use blue light in their laboratories. Thank you.
Thank you, Camilla. Yes, so now we are going to look at some of our customers and how they have used the blue light in their laboratories. So the ones that we are going to have a deeper look at today is Synlab in Estonia, Atria in Finland, Seilab in Finland, and Hankia in Finland. We will start with Synlab. So many of you may know Synlab. So Synlab is a world-leading clinical service laboratory, and this site is in Estonia, in Tallinn. As many of you or many people working in the lab would say to us that they already do disinfection with chemicals, and that should be enough. Synlab also thought so, and they wanted to test if they really need something extra, or if the chemical disinfection was enough for the laboratory benches. The test was conducted in their microbiology laboratory in Tallinn, and the device used was a spectral.blue Salo device. The test setup looked like this. So they had 2 laboratory workbenches and equipment placed on them. And they were first disinfected with chemical disinfection agents just before sampling. And this is how they also do normally every day. After that, they took microbial surface samples with swabs from the working surfaces and the equipment. And these samples were after that cultivated on agar dishes. In the next step, the same workbenches and equipment were illuminated with spectral.blue device overnight. And in the morning, after approximately 12 hours, they again took similar microbial surface samples and from the same locations, and they were also cultivated in the same way as in step 2. What they could see from this small test was that actually when they did chemical disinfection and they thought that their surfaces were perfectly clean, they actually still had quite a lot of microbial burden on the surfaces. But when they did both chemical disinfection and spectral.blue, we could see that they had far fewer colonies on the plates. Conclusions that we can draw from this is that chemical disinfection does not always perform as expected in real-life conditions, and organizations relying only on chemicals are at risk of expensive hygiene failures. And the reason for this is, for example, that the contact times required by the chemical disinfectants to work as they should is between 5 minutes and 30 minutes. Keeping the surfaces moist for this required duration is difficult in practical situations. But with spectral.blue, we can automatically reduce the risk without operator input and can be used also alongside the chemical disinfection. So the best solution is to use both chemicals and spectral.blue. The next customer is Atria in Finland. Atria is a leading food company here in the Nordics. They use spectral.blue Salo XL in their daily work. They have it in their safety cabinet, as shown here in the picture. And they wanted to test how effective the spectral.blue is against Listeria monocytogenes in the biosafety cabinet. They wanted to mirror their routine food quality analysis practices because they wanted to see how it works in real life. To do this, they did prepare the listeria samples in appropriate dilutions, and they had metal discs to which they added the listeria. And 9 of these discs were then evenly distributed within the biosafety cabinets, as shown here in the picture. An additional control sample was placed in a separate cabinet under similar conditions, but shielded from blue light exposure. The 9 discs were treated with multi-wavelength high-intensity blue light overnight for 8 hours from 9 in the evening till 5 in the morning. And this is also how they use the blue light in routine in their daily work. And finally, all the 10 samples were analyzed. What they could see was that after the blue light treatment, all the 9 samples exposed to the multi-wavelength high-intensity blue light were all completely inactivated, with no viable CFU count. But the dark control showed no reduction in the viable CFU count. So the results demonstrate that the spectral blue is highly effective even against challenging microbes like listeria, and can be utilized in biosafety cabinets to prevent cross-contamination and reduce the risk of infection to staff. So they were really happy with the results. Our third customer is a service laboratory here in Finland called SEILab. They are an accredited food and environmental research laboratory. They do regular contamination monitoring in the laboratory and the premises. And in one of these, they had observed that they had Listeria monocytogenes contamination in the waste disposal room. The samples they had were tested with PCR, and they were PCR positives. They did not grow on the plate, but it still were positive in PCR. So they increased the chemical disinfection efforts, but they were not able to fully eliminate the contamination. So that's when they called us and said that they wanted to try spectral blue. What they did was that they placed a Berlin plug-and-play device in the waste disposal room and pointed it towards the known contaminated surfaces. Like here in the picture, you can see that it's pointed towards the autoclave. And after a few days of use, the Listeria contamination could no longer be found. And all this they could not achieve with only chemicals. So as they have seen that spectral blue proved to be an effective solution for decontaminating their premises, they now use it as part of their daily contamination control. And what they also like is that it's safe and that it's portable, so they can also use it in other rooms whenever it's needed or to prevent cross-contamination. Our fourth customer case is from Hankia. Hankia is a leading agricultural products and services company here in Finland. They are part of Danish Agro. They have, as you can see in the picture here on the right, deployed our spectral blue in the ceiling in their grain and feed laboratory. The laboratory is accredited, and they are handling a large number of samples every day from different sources, and they are screening for Salmonella species. The deployment covers the microbiology laboratory rooms and also the equipment maintenance room. And as this is fully automated, they can do continuous disinfection always outside the working hours, and they are using timers and motion sensors. So always during night, the blue light is on. Of course, to be able to show that it works, they do hygiene monitoring in the lab, and they tested the floors, the doors, the switches for the lights in different places, and they have been now able to stop using chemical disinfectants in the cleaning process. And they have also been able to reduce the cleaning schedule from 5 times per week to twice per week. So as they have now noticed, they have been able to to do cost savings, and they have also improved the sustainability of operations by using less toxic chemicals. And what they also have said is that it has made their staff's work easier, as they know that they can always trust the analysis results. And they also know that the health and safety of their staff handling pathogens is safeguarded. So this was 4 of our customer stories. You can find more at spectral.blue. Here we have customer testimonials, both from other laboratories and other sectors. And there you can also see more information about these particular customer cases. The key learnings that we have from talking to these customers is that we have now shown that chemical disinfection doesn't always meet our expectations. Sometimes we need something more. The chemical disinfection process cannot work if full coverage is not achieved and the contact times cannot be obeyed. And this is things that are hard to achieve by doing it manually. That's also why having an automated continuous disinfection is a good choice. Spectral Blue is highly effective against even the most challenging microbes. If we are talking about food and laboratories, service labs, we know that we have a good effect against, for example, Listeria monocytogenes. We can reduce the use of toxic chemicals, which is not only economical benefit, but also good when we think about sustainability. Spectral Blue also makes the laboratory staff's work easier because they can always trust in the analysis results. They do not need to be afraid of cross-contaminations. The health and the safety of the staff handling pathogens is safeguarded. And the fact that the plug-and-play devices are portable makes them quick to deploy and easy to move to another room whenever needed. Thank you. These were the customer cases that we go through today. And now I will hand over to Camilla Höglund.
Thank you, Karin. Still, we don't have any questions in the chat, so we are now opening the ability for you to last minutes to write some questions and ask any of us questions that you may have. While you're thinking and still wondering if there's something you will ask, I will take one of the questions that we get quite often from our customers. So a very typical question is, what about shaded areas or areas that are not directly under the light? So for example, under the table, or in the case of biosafety cabinet, you have areas that might not be directly under the light. And with blue light, we can say that we can quite heavily rely on reflections, because the walls and a lot of our furniture and materials are designed to reflect visible light. And as blue light is visible, it will be reflected from most surfaces. So for example, white painted walls would usually reflect 30 to 50%. And metallic surfaces are really good at reflecting, and they can reflect up to 90% of the light. And in general, blue light is better reflected off surfaces than UV light. So in many cases, and especially since we are offering a disinfection solution that
you can use continuously and many hours during the day. The build-up blue light energy, even in the shaded areas, will be quite significant and enough to provide at least some degree of disinfection. But of course, the best disinfection efficiency you get directly under the blue light. Still, there is no questions coming in the chat, so I will take this time to mention that we still have 2 more webinar sessions coming up this year. So November 12th, we have a webinar focusing on the food industry and food industry customer cases. And the 3rd of December, we have a webinar with focus on healthcare and ambulances. And all these sessions so far we have had, this was the 3rd webinar. They have been recorded, and the recordings of the 2 first webinars are already available, and this 3rd one will probably be available soon to you. So please, you can go and have a look at them afterwards. And if nothing more, I thank you for participating in this webinar, and you are free to send us questions afterwards or contact us afterwards if you have something you want to ask or talk about. And I wish you a really wonderful rest of the day. Thank you.